Hi, everyone. Happy Friday. Oops, sorry. Let me turn on my camera really quickly. All right. Hi, everyone. Happy Friday. My name is Julie. I'm a scientist here at BioTuring, and welcome to this week's Single-cell and Exploration Club. This is where we demonstrate some of the features on our app through the form of a case study. But in particular, this week's club, instead of going through a case study, I actually wanted to go through like a guide or like a tutorial on how to handle GeoMx DSP spatial data on SpatialX. So the agenda for today's club here, what I'll be going through is how to submit GeoMx data on SpatialX and how to look at the data format and actually how to grab that data from the GeoMx platform itself. And then once that data is on the platform, I'll show you guys how you can combine multiple samples, how to perform batch correction, perform the embedding and clustering as well. Then we can go over how to query for genes, visualize those genes on those certain ROIs, perform some ROI annotation. And then lastly, we can also go over how to perform differential gene expression analysis. And at the end, I'll also show you guys how to export some of these charts that you are producing from SpatialX into BioVinci, which is our other platform for customizing these visualizations for scientific publication. All right, so before we even dive into how to handle GeoMx data here on SpatialX, I want to quickly touch on an overview of all of the different types of spatial transcriptomics technology, especially for those of you who are unfamiliar with spatial data or are trying to figure out what kind of spatial technology might be the best for your research interest. So essentially, spatial transcriptomics can be grouped into two different types of technology. The first one is imaging-based, and the second is sequencing-based. For imaging-based technologies, this includes a lot of the popular technologies such as Xenium, Merscope, and CosMx. And these type of technology is really good at getting really high single-cell resolution. So you're able to see the cell boundaries, the cell segmentation, and the cell centers. However, this comes at a trade-off of having a lower gene panel or a lower sequencing depth. On the other hand, you have sequencing-based technologies, so that includes Visium, Visium HD, StereoSeq, and GeoMx DSP here. For sequencing-based technologies, you might not get the single-cell resolution that the imaging-based technologies have. However, these are really good at getting much higher sequencing depth and much higher gene panels, so you're able to analyze and see the expression of much more genes. In particular, for GeoMx DSP, this is kind of unique to other types of sequencing-based technologies because instead of examining individual spots, as you would see with Visium, for example, they're actually examining ROIs or regions of interest. This is where you would pre-select certain regions on the tissue that you'd be interested in analyzing further. And then based on the fluorescence of those regions, you can see what cell types are present within the tissue. So I guess one of the drawbacks to performing GeoMx GSP data is that you would have to know what cell types are already present in your tissue. And typically GeoMx DSP requires or I guess this is a benefit to using GeoMx DSP is that it only requires a small tissue space in order to analyze. So again, the unique thing about GeoMx DSP is that it is a sequencing-based spatial transcriptomics technology with unique regions of interest that you are interested in studying. Down here, I've left the citation to a review article, which I think is pretty helpful in summarizing basically the pros and cons to each of these technologies if you're trying to figure out which spatial technology is best for you. All right, now with that said, maybe we can go over, before we go over the data submission, I do want to show you guys what GeoMx DSP data actually looks like. So moving over here to the SpatialX platform. So here I have my spatial tissue. This is from human brain. And immediately you can see these colored squares. These are the individual ROIs, or regions of interest. So there are multiple regions of interest which you can select on the tissue, and they can look different from each other. So the region can be just a square like this, but it can also look like this. So these regions here are based on the fluorescence of the fluorescent probe. So each of these fluorescent probes will be a marker for a different cell type. If I were to hide the overlay on this particular region, you can see here that the overlay overlaps with the red fluorescence there. And so in this particular study, the red fluorescence here, they used GFAP to mark for, and this is a marker for astrocytes. So not only can you kind of analyze a lot of different cell types together within one region, you can also separate a single cell type within one region as well. Okay, so this is typically what GeoMx data would look like when you load it onto the SpatialX platform here. All right, so let's go over how to actually submit this data to SpatialX. So for the file types that you need, there are two primary files that are output straight from the sequencing machine in order to submit onto the platform here. The first type of file is, of course, an image file. This comes in an omi.tif type of format. And the second file is an Excel file. So this is should be named something like biological probe qc.excel. To actually access these files from the GeoMx DSP program, for each individual image that you open up within the program, you are allowed to export it as an OMI-TIFF format here. So you can imagine if you have different samples or different spatial tissues, then you'd have to open each image individually and then download the OMI.TIFF. Open the next image and then download the OMI.TIFF so that you have multiple images for your multiple samples. For the second file type, the Excel file, you only need a single Excel file for this. So within that single Excel file, it will actually contain all of the information across all of your samples here. So to access that Excel file over here on the right, under the datasets category here, you can see the probe QC Excel file is located here. So this is the one that you actually need to access. Okay, so now that you have your OMI-TIFF and your Excel file downloaded from the GeoMx program, let's go over how the probe QC file actually looks like. So over time, I think the GeoMx program has kind of changed how the export of this Excel file kind of looks like. But in order to successfully submit this type of file into SpatialX, the Excel file should be more or less formatted like this, where you have your multiple samples, as you can see within this first column here, and you also have multiple sheets within this Excel file. So down here in this segment properties sheets, this is where you have across your different samples, you have the marker gene or the fluorescent probe itself. Okay, and then in the target count matrix sheets within that same Excel file, this is where the actual gene expression matrix is located. So it's really important that you have all of these different sheets within one Excel file, because I think over time, sometimes maybe the GeoMx program might actually export these individual sheets as separate Excel files. So it's really important to combine it into one in order to submit it onto SpatialX. One thing that I will note here that is interesting about the GeoMx platform here is that for all of these genes here, you can see there are no genes that have zero expression across all of the regions of interest. This is because the GeoMx machine already does some automatic filtering that will filter out genes that have zero expression in any of the ROIs. So this is important to keep in mind when you perform gene query or gene visualization on SpatialX. And I'll come back to this later to tell you guys why this is important. Okay, and then finally, to actually upload and submit the data here, there are two main ways in which I can recommend you to submit the data onto SpatialX. The first way is by using cloud storage. So of course, a lot of spatial data is going to be very large, so it's highly recommended to have this data already on cloud storage, such as Amazon S3. So you can directly mount that S3 bucket to SpatialX so that you can access your files that way. Otherwise, if you already have your spatial files just downloaded on your local computer, you can also upload it directly as well to the platform through this upload tab here. So those are the two main ways in which you can submit your data onto SpatialX. All right, and so let's go over how to combine different samples together. So once you have your data loaded, it'll look like this, of course, and within this particular data set, I can flip through my different samples over here in the top left-hand corner. In this particular data set, I have 3 different samples, and I want to combine all of those 3 samples together to analyze all of the regions of interest together and all of the different basically ROIs so that I can perform differential gene expression analysis later on in the line. To do so, you'll have to click the Combine Data button here. And for this, I'll name this Combined Samples 1 through 3. For the metadata handling, this is basically asking you if you want to merge all of your metadata from your different samples into one consistent metadata, or if you'd like to keep them separate. I'd like to combine them. And for the technology group, I'll choose nanostring GeoMx, of course. Then I'll select all 3 samples to combine. Okay. And so now over on the right-hand side, it's just a confirmation page saying that you want to combine all of these 3 samples together. And so once you've combined these, what you'll get is something that looks like this. So now you see all of my 3 brain regions combined into one view. And then across all of these different brain regions, I also have the different ROIs that I can see. And when I combine the metadata into one, I can also see my ROI labels combined into one metadata category. Okay. All right, so now that covers how to combine samples with the metadata. So let's go over how to perform embeddings and batch correction. So for embeddings, how to access that tool is you actually want to switch the display over from the image view to the 2D view up here in the top toolbar. Doing so, you'll be able to see these tiny spots. Let me up the point size so you guys can see a little bit better. So each of these spots here represents a separate ROI. You can see that they're colored differently, and the ROI label over here represents the ROI number. And up here, you'll also notice that this says 173 cells, but for GeoMx data in particular, each of these individual dots actually represents an ROI. So you want to interpret this as 173 separate ROIs. Now to perform an embedding, you can click open the embedding tool up here in the upper left-hand corner. We can choose PCA as the embedding. And for the normalization method here, it's really important to choose either the raw counts or the log normalized counts here. Maybe these scaling here, it won't be appropriate for the embedding because basically, as you can imagine, for log transformation or for log scaling, essentially what that does is it kind of takes your gene expression values, the range of gene expression values, and squeezes it into a smaller range. And so in doing so, it kind of might, you might not be able to see the differences between the gene expressions of certain ROIs if it's squeezed or transformed in that way. So in that respect, again, the normalization method that is typically accepted for GeoMx DSP data is either the raw counts or the log normalized here. Finally, in this batch key option, this is where you can perform a batch correction. So you want to choose something like the default batches information, batch ID, sample ID, subject ID as the batch key to perform the batch correction on. And once you run this, I've already run a result here. It'll change the dots here. So now you can see your spread of your individual ROIs on a separate embedding. I can also display the X and Y axis here if you guys want to see the principal component axes. So now this will allow you to see how your different kind of ROIs are clustered and spread across principal component axes like this. All right, and so to summarize, you can create new embeddings for your regions of interest and also perform batch correction. So in particular, this performs batch correction with SCVI automatically. All right, so now that we've performed embedding, I think the next natural step here is to perform clustering. So clustering in single cell data, for example, when your single cells are clustered together, certain cells will be clustered in one cluster depending on how similar their gene expression profiles are. So the same kind of methodology can be applied for clustering on certain ROIs. However, one thing to keep in mind for the clustering here is if you were to perform the Louvain clustering, for the input embedding, you want to use the embedding that you just performed in the previous step. And underneath the advanced parameters here, you'll notice that we set the default number of neighbors to 90 here. And now for GeoMx data, you want to set the number of neighbors here equal to the number of ROIs that you have within your sample or less. So some of you within your samples might have only 50 ROIs. Then in that case, you want to set this number to 50 or less. But in my case, since my number of ROIs is 173, then I can leave it at 90. Okay, so keep this special consideration in mind when you're performing the clustering analysis here. All right, so that covers clustering now on GeoMx DSP data. Now I can also show you how to subcluster some of your samples here. So for some of you, you might have samples from different diseases or different conditions. Let's say, for example, this ROI number 1 is like my control group, and the rest of my ROIs come from tumor tissue. So what I can do is I can separate my tumor tissue from the control group by selecting Shift or Control on my keyboard, holding that down, and then selecting all of my regions of interest or my metadata label, so that now down here, you can see that I have 171 ROIs selected. From here, I can subcluster this. Okay, and so now I'm only looking at my tumor tissue, for example. I can also choose to save this cluster, subcluster as tumor tissue. Okay, and so once you've done so, whenever you subcluster, please remember to also reperform the embedding and reperform the clustering as well, because now you've separated some ROIs from each other. So now you want to reperform the embedding on that. And so essentially, this is what these slides cover, is that when you select a subcluster, then you can save that subcluster and then also remember to rerun the embedding and the clustering after you have subclustered your ROIs. All right, this is to rerun the clustering analysis here. Okay, and so let's go over some gene query visualization and how to annotate some ROI regions based on the gene visualization. So let's go back to all of my ROIs here. And maybe it'd be more interesting to look at the spatial tissues here. So let's say I'm interested in some 2 random genes, this gene and this gene here. and I want to look at the expression of both of these genes. What you'll notice immediately is all of the ROIs express both of these genes. And this is going back to that previous slide during the introduction, where I mentioned that the GeoMx machine does some automatic filtering to the genes so that it will remove genes with zero expression. So any gene that you query with other genes will automatically always co-express with the other genes. And so maybe this co-expression tab might not be the best way to look at how two genes are co-expressed with each other, because all of these ROIs will always co-express these two genes together. Instead, you can look at the AU cell enrichment of these two particular genes. So essentially what this will do is it will calculate an enrichment score that will tell you how enriched this gene set is within your ROI. And you can see here how a certain population of ROIs expresses the AU cell enrichment more highly. So if you were to select along that population of high AU cell score, now you can see I'm selecting 29 ROIs, and now I can annotate these ROIs as having, let's see, AU cell enrichment, and this has high AU cell enrichment. Okay, and that's how I can annotate my ROIs and create new metadata over here on the right-hand side to refer back to it whenever I need to. Okay. All right, so that covers how to perform some gene query here on SpatialX, and then also how to annotate your ROIs here. Okay, again, it's recommended to use AU cell scores if you want to look at the enrichment of multiple genes together. All right, then finally, let's go over some marker gene and differential expression analysis here. One thing that I will mention here first is that for both marker genes and differential expression analysis, the algorithm behind those two methods are exactly the same on the back end. So you'll notice how in marker genes, we have methods like t-test, Venice, and Wilcoxon. These methods are also available in differential expression analysis, but the reason why we separate these two tools is because it will give kind of different visualizations based on, kind of the results that you are expecting. One thing to keep in mind that is very important is that among these three methods, the most appropriate methods for GeoMx DSP data is particularly the t-test or the Wilcoxon method. This is because the Venice method is more optimized for single cell analysis. And of course, we don't have the single cell resolution that we do in GeoMx data. So again, please choose the t-test or the Wilcoxon method for marker gene analysis and differential expression analysis here. To demonstrate that on SpatialX here, we can select certain ROIs that we are interested in. For example, I can use my little lasso tool down here, and I can select these three ROIs. Okay, and now down here, you can see that I can run the marker gene analysis, run that. And what that will do is over on the right-hand side, you should be able to see a list of genes that are the marker genes for these three ROIs. Okay. And so now we have our list of marker genes here. And so you can add these marker genes, to the gene query panel on the left-hand side if you want to see kind of the expression or the AU cell enrichment of these genes within this particular ROIs there. Okay, and then the same can be done for differential expression analysis as well. So for differential expression analysis, again, you can choose the t-test, and this is where you will choose your two populations of interest to kind of compare. So let's say I want to compare my ROI labels. Let's say ROI label number 1 versus ROI label number 2. So And then I can run an analysis. Again, it's going to give you a list of differentially expressed genes in a table format on the left-hand side. And then once it loads, you will be able to see a volcano plot as well. Okay, something like this. So while we wait for the kind of genes to load here, really quickly, maybe I can just cover the results in this slide. So here again, you can perform DGE analysis using the t-test or the Wilcoxon, and then choose your two groups to compare against in the differential expression analysis. All right, and so the last thing that I want to cover here is actually BioVinci. So you'll notice that we have a charts and graphs tool within SpatialX here. And within the basic charts, you can build some of these charts for publications such as violin plots, box plots, bubble heat maps, or upset plots. So for example, if you were to look at certain genes, the gene expression across your different ROI labels. So let's take a look at that. Okay, and now I'm looking at the expression of these certain genes. Up here, you'll notice that we have an export to BioVinci button. So what that will do is it will export these violin plots over to BioVinci. It's going to open up a new tab in BioVinci, so make sure to have your pop-ups enabled on your browser. And then to save time, let's just switch to BioVinci tab that I have open here. So you can see here in this violin plot, some of the things that I can do is over on the, oops. So over here on the left-hand side, this is where you can change the different colors. You can also kind of reorder these violin plots as well. You can do things like change the font size, change the font type, the font colors, essentially prepare it to make it ready for your publication. And then lastly, you can export it into many different high quality formats. So let me show you here after this is done saving. Okay, maybe to save time, basically I can just list out the chart options. Okay, here. So the file formats that you can export in high quality are PDF, SVG, PNG, and JPEG. You can also choose the resolution to be a little bit higher if you'd like, and the color mode as well. Okay, so feel free to play around with this BioVinci application here. This BioVinci application is also connected to our other apps such as bbrowser X Pro and SmartBulk as well. So anytime you see the little export to BioVinci button here, you're allowed to export that into BioVinci and then make some further customizations. All right, and so that is it for today's Single-cell and Exploration Club. So thank you everyone for attending this week. In a little bit, my colleague is going to open up the poll here. So we'd be interested in getting more feedback on SpatialX, see what you guys are interested in learning about next. And of course, we always welcome any other feedback that you have for SpatialX or for this webinar series in general. Okay. So I'll give it maybe a minute or two for everyone to finish filling their poll. Otherwise, we are also open to Q&A at this time as well. So let me check the Q&A. Okay, so we have one question from someone who asked, does similar workflow apply for 10x Visium data analysis in terms of embedding and clustering? I would say yes, the workflow is going to be very, very similar. But maybe if you're interested, we can also make a separate kind of guide or tutorial on handling Visium data and how to submit the data onto SpatialX as well. But yes, generally the embedding is going to be the same. You can perform PCA. Clustering is going to be the same as well. Just keep in mind the number of neighbors as well. Okay, thank you. Another question from someone asks, hi, I'm working on MERFISH spatial transcriptomics data. Can I upload two different samples, two different files, and then perform downstream analysis together? So yes, you can. So I can demonstrate that really quickly on the app here. So if you were to add a new study on SpatialX here, you'll have to switch the platform over to MERScope. So we have MERScope version 1 and 2. So make sure to specify which one you have. And the file types that you would need for submission are, of course, the image files, the segmentation files, and then the transcript files. So primarily those three files. And let's say you were to enter this one sample. So sample 1, you would drag and drop the files here. And then you would click open new sample, sample 2, to drag and drop the files there. And then in order to combine these two samples together, you'll have to do the combine data that I demoed a little bit earlier. So down here, this is where you can combine the data. So you see here, I have my different samples so that I can analyze them separately. Or if I want to analyze them all together, I can also combine the data here as well. All right. So if that answers your question. Okay, last question here. Someone asked, what about if data sets are downloaded from publications? So that's completely okay too. There's kind of two approaches in which you can take for this, take to this. So the first approach is if you download these publications yourself. So the authors have already provided all the files. You download it yourself. You can also submit it yourself to SpatialX. That's completely okay. The second way is also by requesting studies. So if you log on to the Bioturing ecosystem here, we have a request management page. And this is where you can request our data team to curate certain studies to put on Talk2Data, which is our database for single-cell, spatial, and bulk RNA sequencing studies. What you can do here is you can add a new request, and all you have to do is input the name of the paper and, more importantly, the link to the paper. And our data team will get back to you to see if the authors have provided enough files and data sets in order to curate this and put it on our database. And so our turnaround time is about 2 weeks or so, depending on our workload. But after that time, you will receive an email with an early access link. So with that link, you can open up that data set in SpatialX and then continue exploring from there. Okay. Okay, and so another question here, if I upload author's annotated data set, will that require embedding and similar workflow? So this actually is kind of dependent on the file type that the authors have provided. So for example, sometimes the authors provide the pre-processed kind of data already, for example, in an ANData format. Sometimes within that ANData file type, they already have performed the embedding for you. So when you submit the data to SpatialX, you can actually access that embedding already because it should have been already imported upon submission. Otherwise, you can also just rerun the embedding based on your own parameters, or if you follow the author's parameters as well, you can rerun that embedding and perform the workflow from there. Okay. All right, so thank you, everyone. I think this has been a really productive webinar this week. A lot of questions so far. So if you have any further questions, feel free to contact us at support@bioturing.com. If you want to schedule a one-on-one, ask questions via email, or if you don't yet have access to Bioturing, you can also request a free trial as well by reaching out to us at that email. Okay. Right. Thank you, everyone, for joining this week's Single-cell and Exploration Club. We hope to see you back in 2 weeks. All right. Bye.